Hello everyone, today we are going to start one topic that is called the mechanism of enzyme action. So before going in detail, let me tell you about that for any chemical reaction to occur, the reactants have to be in an activated state, right? Or that is called the transition state. So the enzymes generally it enhance the rate of reaction by lowering the activation energy and by forming the enzyme substrate complex. So what is the meaning of activation energy? So generally most reactions they do not start automatically. Okay, the reason is the reactions have some energy barriers to become reactive, which is maybe due to the hydrogen bonding or the absence of process collisions or because of the mutual repulsion. So the external supply of energy needed to overcome this barrier is called the activation energy. Okay, so step is the formation of the enzyme substrate complex, right? So there will be formation of the enzyme substrate complex. The enzyme will binds with the substrate at the particular site, that is active site, and form the enzyme substrate complex, which ultimately results in the production of product. Okay, so the active site of enzyme have specific conformation for attracting and holding substrate. It usually possesses a pocket-like structure where the substrate fits in the complementary fashion, that is the active site, and they two join to form the enzyme substrate complex. Okay, and the enzyme substrate complex, then it is a very short-lived complex. The substrate is changed into enzyme product complex. Then the product remains complex within the active site of the enzyme for a brief period, and it soon separates, and the active site is free to perform another catalytic act. Okay. So the enzyme combines with the molecules and bring them close together, which favors their collision in the most suitable directions and locations for the reaction to occur. Okay. So the next thing is we'll discuss about the transition state. Okay. So what is transition state? As I already told, transition state is the activated stage. Okay, so lowering of activation energy by enzyme and the energy reactions of the chemical reactions. Okay, so as I said, to overcome the energy barrier there is the need of activation energy. Okay, and what is the activation energy? It is the external supply of energy needed to overcome this barrier. So you can see in the x-axis the progress of reaction, enzymatic reaction, and the y-axis there is the potential energy or the energy coordinates. And you can see this is the substrate, and this is the transition state, and this is the formation of the product. Okay, so the enzyme lowers the activation energy of the reaction and allows a large number of molecules to react at a time. Okay, so you can see in this case when the activation energy is more, there is non-enzymatic reaction, and in this case as the activation energy is less, this is a enzymatic reaction. That means the enzyme enhance the reaction by forming here the enzyme substrate complex by lowering the activation energy. Okay, so you can see for the enzymatic reaction the activation energy curve is low, and in this stage the non-enzymatic reaction the activation energy curve is more. Okay, so this is about the transition state or the activated state. Okay, so the next is about the two hypotheses. That is, one is lock and key hypothesis, and second one is about the induced fit hypothesis. So this is called as the lock and key hypothesis. So in the case of lock and key hypothesis, it was put forward by Emil Fischer. Okay, so according to this hypothesis, both the enzyme and the substrate molecules are specific geometrical safe. Okay, and in the region of active site, the surface configuration of the enzyme is such as to allow the particular substrate molecule to be held over it. Okay, so the active sites also contain special groups like NH2 groups or COH groups or SH group for establishing contact with the substrate molecules. The contact is such that the substrate molecules or the reactants come together, causing the chemical change. It is similar to the system of lock and key. Just as the lock can be opened by a particular key or the specific key, a substrate molecule can be acted upon by a particular enzyme. Okay, and then it form a enzyme substrate complex. So in the complex stage, the molecules of the substrate undergo the chemical change and then the product will be formed. Okay, so this model doesn't give any scope for flexible nature of the enzyme, hence it fails to explain many facts of the enzymatic reactions, and the most important being allosteric modulators. Then the next is about the induced fit hypothesis. So the induced fit hypothesis, it was proposed by Daniel E. Goland in 1959. So according to this theory, the active site of the enzyme contains two groups. Okay, so we have already discussed the active site, and the active site, it contains two groups. Here you can see this is the buttresing group and this one is the catalytic group. Okay, so the buttresing group is meant for supporting the substrate. Okay, the buttresing group is particularly meant for the support of the substrate, and the catalytic group is able to weaken the bonds of the reactants by electrophilic and the nucleophilic forces, and the two groups are normally at a distance. You can see this is the buttresing group and this is the catalytic group, right? So as soon as the substrate comes in contact with the buttresing group, the active site of the enzyme undergoes conformational change. Okay, so as to bring the catalytic group opposite to the substrate. Okay, so this is the substrate. When the substrate comes in contact with the buttresing group, the active site of the enzyme undergoes a conformational changes so as to bring the catalytic group exactly opposite to the substrate. So the catalytic group helps in bringing about the chemical reaction, and the substrate is converted into the product, and the product is unable to hold on the buttressing site due to change in its structure and bonds. So the buttressing group reverts to its original position, and after that only the product will be released. Okay, so first there is two groups, one is buttressing group, one is catalytic group. So the buttressing group is for the support of the substrate. So when the substrate comes and binds to the active site, the contact with the substrate comes in contact with the buttressing group, the active site of the enzyme undergoes conformational changes. Okay, and after that it brings the catalytic group opposite to the substrate. So the catalytic group helps in bringing about the chemical reaction, and the substrate is converted into product. Okay, so then the product is unable to hold on the buttressing group due to the change in its structure and bonds. Okay, that is already the structure and conformation has been changed because of the attachment of substrate. So when the buttressing group reverts to its original position, then only the product will be released. Okay, so the induced feed model has sufficient experimental evidence from the X-ray diffraction studies, and it also explains the action of the allosteric modulators and the competitive inhibition of enzymes. We'll discuss later about the modulators and the inhibitors. Okay, so the next thing is about the differences between the lock and key theory and the induced feed theory. So in the lock and key theory, the active site is a single entity. Okay, but in induced feed theory, as I already mentioned, the active site is made up of two components. One is the buttressing group, one is the catalytic group. So in the second point is there is no separate catalytic group, but here is a separate catalytic group. And the third point is the active site is static. Okay, and in the induced feed, the active site is not static. Fourth step is the development of transition stage is not considered, but in this case it considers the development of transition state before the reactants undergo change. The fourth step is does not visualize the weakening of the structure substrate bonds. In case of induced phase, the catalytic group is believed to weaken the substrate bonds by nucleophilic and the electrophilic substitution. Okay, and the last step is it doesn't explain the mechanism of non-activity in case of competitive inhibitor, and in induced field it explains the mechanism of the non-action over competitive inhibitor. Okay, so this is all about the mechanism of enzyme action, how it lowers the activation energy and forms the activated stage or the transition state and results in the formation of product. Then there are two hypotheses, that is, one is lock and key theory and one is induced phase theory. So this is all about the mechanism of enzyme action. If you like this video, hit the like button, share it with your friends, and do not forget to subscribe to this channel. Thank you.